Okay, introducing our next speaker, Nazia Thakur from the Purbright Institute.
Okay, great. Hi, guys. I'm going to talk to you about some of the work we've been doing to examine the antigenicity and host range of SARS-CoV-2 variants and other bat sarbecoviruses. And some of this work was funded by the Pump Priming Award that Julia mentioned this morning. So thank you to the network for that. And we're in the process of currently writing up and submitting the manuscript for this as well. So there have been several bat sarbecoviruses that have been isolated across the globe, and these are phylogenetically clustered into different clades, with the clade 1, 2, 4, and 5 viruses found primarily in parts of Asia, and the clade 3 viruses in parts of Europe and in Africa. And the bat species that these viruses have been isolated from are also quite widespread globally, with the highest density of these bat species being found in Asia, suggesting that there could be potential for these viruses to spill over. So, for the purpose of our study, we've taken representative sequences from each of the different clades to understand what their spillover potential could be for these viruses. So, what we want to do is understand what the potential is of these bat sarbecoviruses to spill over from their own hosts, and then what's the potential of them to infect other hosts, and then use this information to understand what the risk could be to other animals that could act as intermediate hosts for the virus, as for example we've seen with SARS-CoV-1 using the palm civet. And then we can then understand what new animal reservoirs they could be from these viruses. So as was seen with SARS-CoV-2 and infection of white-tailed deer and the American mink. So to do this species tropism assays, we have taken libraries of bat sarbecovirus spikes, use them to generate pseudoviruses, and because these constructs are tagged, we can check for expression in purified virus. And we've got a separate library of around now 50 bat and mammalian ACE2 constructs that again are tagged and we use to overexpress in mammalian cell lines and again can check for their expression. So what we've then done is taken these two libraries and either generated purified proteins to look at binding and affinity, or use them in pseudovirus entry assays to look at host range. So if we first have a look at the sort of conservation of these different bat sarbecoviruses, this is the RBD of SARS-CoV-2, and we've compared it to the sequences of the other bat sarbecoviruses that we're looking at. And you can see that at the ACE2 RBD binding interface, that actually looks like there's quite a lot of divergence in the sequences. And if we look more closely at those sequences, the only thing really you need to take from this is that within the different clades, there are areas of deletions within this binding interface, suggesting that there probably is some difference in species tropism of these viruses as well. So what we then did was we took the ACE2 that we had of the bats, overexpressed them in mammalian cells, and then infected them with the different sarbecovirus pseudoviruses. And what we saw was that with the clade 1b viruses, which is what SARS-2 is part of, we saw this broad generalist ACE2 using phenotype of the bat receptors that we had. And then the clade 1a viruses had still a generalist phenotype, but there was some evidence of restriction in this ACE2 usage. The clade 3 and 5 viruses were more restricted and showed a more specialist phenotype, with these viruses only really being able to use the cognate bat ACE2 receptors that they were isolated from. And then the clade 2 viruses didn't use ACE2 at all, which is already well established. So what we were also able to do is correlate our entry data to binding in either ELISA or by flow cytometry, where we saw this similar pattern of generalism and specialism between the different clades. So the question we then had is, okay, well, if these viruses are able to use different bat ACE2s, what's the potential of them using sort of larger species ACE2s like from different mammals? So our previous work has shown that SARS-CoV-2 has a broad tropism for mammalian ACE2 with some notable restrictions in rodents, birds, and some bat species. And this was particularly important when new SARS-2 variants emerged, because we actually then saw that there was a shift in receptor tropism, which others have shown as well. And we were also able to pinpoint the sort of key determinants of this switch in tropism to mutations in the spike protein positions 501, for example, in alpha for mouse and rat ACE2. So what does this species tropism then look like for other mammalian ACE2s with the bat sarbecoviruses? And actually, it looks like the tropism looks quite broad compared to the bat ACE2s, where it seemed more restricted, where particularly the clade 1 viruses are able to use all these mammalian ACE2s. So we've tested sort of livestock and pets, rodents, suggested intermediate hosts, and then some monkeys. And the only restriction we really saw was in this new world monkeys. But we did see some restrictions with viruses like RATG13, again, the clade 3 viruses. And then what was quite interesting was that the clade 5 virus, RCO319, didn't use any of these mammalian ACE2s at all. So this suggests that when we're thinking about maybe predicting spillover of viruses, actually where they're based in their clades might not be the be all and end all, because you do see these outliers like we've seen for RATG13. So most of this work was done with just the wild type SARS-CoV-2, but we wanted to understand what would this look like in a post SARS-CoV-2 emergence era with the different variants that emerged. And I'm just focusing on the bat ACE2s because actually for the mammalian ACE2s, it looked like there was quite broad coverage like we'd seen previously. But for the bat ACE2s, what we saw was that for the earlier variants like alpha, beta, and delta, there seemed to be like a similar pattern of bat ACE2 usage, with some of these variants having restricted bat ACE2 usage initially, but then with Omicron were able to be used by this virus. Or we saw the opposite phenotype where initially they were used by the different variants and then lost this phenotype. So what we wanted to have a look at again was what genetic determinants could explain why we saw this shift in tropism with the variants. So we built point mutants into a B1 background to make it more BA1-like, focusing on mutation at positions 417, 484, 493, and 501, which have been described as being important in receptor tropism. And we saw that particularly when we had 493, either by itself or in combination with other mutations, we saw this shift in phenotype that we'd seen for BA1 down to Omicron. So the next question we asked is, okay, so these viruses seem like they have tropism for mammalian ACE2s, but actually a lot of them had tropism for human ACE2. So if these viruses were then to ever spill over into the human population, would the pre-existing immunity we had against SARS-CoV-2 provide any protection against these other viruses? So to look at this, we took a panel of convalescent sera from individuals who had been infected with different SARS-CoV-2 variants and then looked at their neutralization phenotype. So just to note, for RHGB07 and RC309, because they don't use human ACE2 so well, we used their cognate ACE2 receptor from the bat that they were found in. And what we actually saw was that most of these viruses seemed like they were neutralized by all of the convalescent sera that we tested. And looking at it a bit more sort of broadly, that if we combine all that sera, they show a similar phenotype, with the ones that had sort of this broad generalist ACE2 usage also neutralized the most strongly, whereas the ones that had a more restricted ACE2 usage were still neutralized, but not as well. But it does maybe suggest that if these viruses were to spill over, there could be some potential of pre-existing immunity providing some degree of protection. So to look at this a bit further, we've taken a panel of monoclonal antibodies to understand what the sort of epitopes are that can serve between these different sarbecoviruses. And these monoclonal antibodies are from Katie Dawes' lab at King's College London, who has, with her team, isolated monoclonal antibodies from breakthrough infections from SARS-CoV-2 individuals. And these map to different regions of the SARS-CoV-2 RBD determined by competition ELISA with known monoclonal antibodies. So what we've seen, so this is for some binding data, that similar to SARS-CoV-2, the banal viruses, which is like a part of clade 1b, were able to bind strongly to all of these monoclonal antibodies. But when we looked at some of the other sarbecoviruses, like the clade 1a, 5 and 3, you see a dropout in these in this binding. But what we do see is that there are specific monoclonal antibodies from group 2, 3, and 4 that seem to be binding all of these RBDs. So this is some work that we're doing with Katie's lab to try and determine what the sort of conserved epitopes might be for this sort of broad binding of these monoclonal antibodies. So just to summarize, I've shown you that the sarbecoviruses, they show broad species tropism for both bat and mammalian ACE2s, but there's some obvious restrictions that we see with the sort of more specialist clade 3 and 5 viruses. We see that, again, similar to the tropism, we see that the generalist ACE2 users seem to be able to be neutralized better by convalescent sera and also bound better by monoclonal antibodies. But we do still see some cross-neutralization and binding with the more distant and sort of more restricted sarbecoviruses. So some future direction that we're taking, I've already mentioned, but we're trying to determine what the key residues are that are involved in species tropism, but also in the antigenicity that we're seeing. We've sort of touched upon this, but like, can we use it to predict spillover, particularly if we're seeing some outliers in our tropism studies, and what other determinants could there be of host range, for example, like cellular proteases that are involved in entry. And with that, I'd just like to thank everyone involved in this work, particularly Joe Newman, who did most of this work with me. And thank you all for listening.
Amazing. Thank you for a great talk. Have we got any questions?
Can you help us out from in the room?
Just out of curiosity, so this system that you've developed to screen spikes for binding ability, how rapidly deployable would that be, for example, in the context of a novel emergence?
Yes, I mean, they're quite quick because you can just get the construct synthesized, and we're just based on the spike protein. And if we're thinking about it in terms of like the pseudovirus system, for example, like we train people quite regularly to come and set that up in their own labs, and it's just a transfection based assay, so it's quite simple. And the same with, like, for example, the ACE2s, we now have them like readily available. If anyone ever wants them, we just like send them to them. So yeah.
Time for one more question.
Can I ask a question? So in terms of the ACE2 expression, as I said, is a transfection-based, how physiologically relevant are the level of ACE2 that you're getting on your cells compared to what would maybe expect in
plants? So they are, of course, like, it's an overexpression because we want to see if we can see that entry or not. But I think what's important is that for viruses that you don't see any entry, even if you're overexpressing, you just don't see anything. And with some of them, for example, with like the SARS-2 variants, we normalize them based on, like, say, human ACE2 expression. So you really do know whether you're seeing an increase in entry or not.
Okay, cool. Thank you.
Amazing. Thank you very much.